Welcome to today's lecture on cell biology, specifically focusing on various aspects of cardiovascular physiology. Understanding the intricate workings of cardiomyocyte development, cardiac muscle contraction, cardiac remodeling, and other essential topics are crucial for medical professionals in providing effective patient care. In this script, we will explore these topics in detail, addressing their importance and mechanisms. So, without further ado, let's dive into the fascinating world of cardiovascular cell biology. In the cardiomyocyte development section of Cell Biology of the Cardiovascular System, we explore the remarkable process through which specialized heart muscle cells, known as cardiomyocytes, develop. This involves complex signaling pathways, morphological and functional changes, and the expression of specific transcription factors. Understanding this development is crucial for identifying and treating congenital heart diseases. Cardiac muscle contraction is a crucial process that pumps blood throughout the body. It involves complex events like depolarization and repolarization of cardiomyocytes. At the cellular level, the sliding filament model explains how actin and myosin filaments interact to produce contraction. The electrical conduction system ensures coordinated contraction of the heart chambers. Understanding cardiac muscle contraction is vital in diagnosing and treating heart conditions. Cardiac remodeling involves changes in the structure and function of the heart in response to injury, stress, or disease. This includes hypertrophy of cardiomyocytes and alterations in the extracellular matrix, leading to cardiac dysfunction. Potential therapeutic strategies can be developed by understanding these mechanisms. Endothelial cells line the inner surface of blood vessels and play a crucial role in maintaining vascular homeostasis. These specialized cells regulate vascular tone, control permeability, and modulate inflammatory processes. Endothelial cells continually release nitric oxide, NO, a potent vasodilator that helps regulate blood flow and maintain vascular tone. Additionally, they express adhesion molecules, such as selectins and integrins, which facilitate leukocyte recruitment during inflammation. Furthermore, endothelial cells secrete various vasoactive substances, including endothelin-1, prostacyclin, and thromboxane A2, which regulate vascular smooth muscle tone and platelet aggregation. Understanding the intricate interactions between endothelial cells and the vascular environment is crucial for addressing cardiovascular diseases such as atherosclerosis, hypertension, and thrombosis. Smooth muscle cells, SMCs, in blood vessels are important for controlling vessel diameter and blood pressure. They contract like cardiac muscle, which constricts the vessels. SMCs also create and maintain the structure of blood vessels by producing collagen and elastin. Knowing about SMCs is essential for managing conditions like high blood pressure. Platelets, derived from megakaryocytes, help maintain hemostasis and prevent excessive bleeding by adhering to injured blood vessels and releasing factors that aid in clotting. They contribute to clot formation through shape change and formation of a platelet plug. Understanding platelet biology is important for managing bleeding and clotting disorders. Cardiomyocyte excitation contraction coupling is the process by which action potentials trigger intracellular calcium release, leading to muscle contraction. This complex process involves several key proteins and ion channels. The action potential propagates along the sarcolemma, the plasma membrane of cardiomyocytes, resulting in the activation of L-type calcium channels. Calcium influx through these channels triggers the release of calcium from the sarcoplasmic reticulum through the ryanodine receptor. Increased calcium concentration within the cytoplasm binds to troponin, initiating the cross-bridge cycling that leads to myofilament sliding and muscle contraction. Understanding the excitation-contraction coupling process is crucial for comprehending cardiac electrophysiology and the development of therapeutics targeting arrhythmias and contractile dysfunction. Cardioprotection aims to prevent or minimize heart damage during ischemia and reperfusion injury. Strategies involve promoting cell survival, reducing oxidative stress, and preventing apoptosis. Signaling pathways, like PEAK/ACT and ERK1/2, activate prosurvival proteins, suppress cell death pathways, and enhance antioxidant defenses. Mitochondria are crucial for cardioprotection, maintaining health, preventing pore opening, and facilitating ATP production. Studying these mechanisms can lead to therapies that minimize damage and improve patient outcomes. In the field of cell biology, the process of angiogenesis and neovascularization plays a vital role in tissue perfusion and wound healing. Factors like VEGF, FGF, and angiopoietins promote angiogenesis, while angiostatin and thrombospondin-1 inhibit vessel growth. Neovascularization in ischemic tissues relies on endothelial progenitor cells and growth factor pathways. This knowledge helps in developing therapies for conditions like limb ischemia, myocardial infarction, and stroke. The concept of cardiac stem cells has revolutionized our understanding of cardiac regeneration and repair. These cells possess the ability to self-renew and differentiate into various cardiovascular cell types, including cardiomyocytes, endothelial cells, and smooth muscle cells. Cardiac stem cells reside within specialized niches in the heart, such as the epicardium and endocardium. Upon injury or disease, these stem cells can be activated and mobilized to promote tissue regeneration. Harnessing the potential of cardiac stem cells holds immense promise for myocardial regeneration and functional recovery following cardiac injuries. Ongoing research aims to optimize their isolation, expansion, and delivery strategies to translate these findings into clinical therapies. Cardiac fibroblasts are important cells in the heart that have recently been found to play active roles in cardiac development, remodeling, and repair. When the heart is injured or under stress, these cells transform into a myofibroblast phenotype and produce more extracellular matrix proteins, like collagen. This remodeling process is necessary for scar formation and wound healing, but excessive fibrosis can impair heart function and lead to irregular heart rhythms. By studying how cardiac fibroblasts interact with other heart cells in the extracellular matrix, we can identify potential treatments to modulate cardiac remodeling and promote tissue regeneration. Cardiomyocytes have an efficient metabolism, primarily using fatty acids and glucose for energy. Fatty acid metabolism generates ATP through beta oxidation, while glucose metabolism provides fast energy through glycolysis and the tricarboxylic acid cycle. Dysregulated metabolism can impair cardiac function, requiring targeted interventions for optimal energy utilization and cardiac health. In conclusion, the study of cell biology within the cardiovascular system offers valuable insights into normal physiology and diseases. Topics covered, such as cardiomyocyte development, cardiac muscle contraction, and endothelial cells, are essential for medical professionals to effectively manage cardiovascular diseases. Thank you for joining me in this exploration.